441524. That's what it converts in the body. It's an adenosine analog. It reduces the viral reproduction. So it's a viral replication of the memory RNA is getting blocked. It's not a rapid RNA producer messengers. It interferes with the RNA-dependent RNA polymerase, which replicates so fast in this particular virus. It evades proofreading by viral exonuclease. So the virus is again a rapid supercomputer. So it starts to read what is need to be read to replicate itself. Simple understanding. So this evades proofreading by viral exonuclease. GS441524 has shown activity against SARS-CoV-2. That is a good message. So remdesivir to GS441524 from prodrug position to a drug position and incorporates into new viral RNA strand, very vital, and polymerase enzyme stops adding more RNA subunits. Hence, it's hitting right at the replication phase. The mode of action of ivermectin. This is an antiparasitic drug which came out to prominence in Australia. It binds selectively to glutamate-gated chlorine channels. So it binds selectively to glutamate-gated chloride channels in invertebrate muscles and nerve cells. From there, it causes hyperpolarization. So antiparasitic drugs works on the cell membranes and the cytoplasm, which we are going to talk later. And it causes paralysis and death. So hence, some broad-spectrum antiviral activity in vitro or in vitro against SARS-CoV-2 is being noted. Mode of action of hydroxychloroquine. Now, this is another vogue drug which has been highly publicized in France, the one which came out very early. And Nobel laureate Arnold, he was raising this as a mode of action of hydroxychloroquine were highly spoken in France first by the Nobel laureate Arnold. There have been positives and negatives and various views. There are some of the head of states as also endorsing it. You can see across the pond there's a great words about it, and hydroxychloroquine is widely spoken. We do know that it's an anti-malarial drug and also it's been used in autoimmune diseases. I'm not talking about the autoimmune SLEs and about the rheumatoid arthritis at this stage, but we do know about this drug is existing. But I'm just going to talk about the mode of action of the hydroxychloroquine. If you may go back to read about Harrison's textbook of medicine, you will see many years ago, as we all read, one of my colleagues, Professor Ravindran Ranjit, he actually showed this to me, and it's a very valuable highlight by him. And I think you all should go and read, and you must read that chapter. There will be questions on the hydroxychloroquine. You got to know in and out of that. It accumulates in the endosomes and lysosomes. It accumulates in endosomes and lysosomes. Lysosome is the one which, and the lysosomal effect is the one which is called the enzyme which come and hits and kills them, which is called the killing effect enzymes within the cells and endosomes. So this particular medication of the drug, it increases the pH in the antigen presenting cells, that makes it to basophilic rather than acidophilic, which they can replicate. And it also causes stabilization of lysosomal enzyme activity, thereby making the lysosomal enzyme very strong and will be able to have a very effective, very tight of the borders or the walls of the lysosomes. And trapping free radicals, suppression of T lymphocyte cells. You must know the T and B lymphocytes by heart. So antiviral action is unknown. Reduced acidity, as I told you, in endosomes and prevents release of virus into cytoplasm. That's important. In vitro activity against virus, but not translated in animals or humans. Well, these are also early stage. Everybody is a scientist at the moment. Lots of scientists have different versions. We are waiting for the outcome and the policies on this. Then comes the mode of action of TNF inhibitors. This is very important to understand tissue necrosis factor. You would see in your own hospitals where working, the protocols are there to using of the TNF inhibitor. These are the ones which is used in the autosomal, autoimmune diseases and etc. The TNF made by activated macrophages. You must know that. There is a slide coming with a good picture. You will be able to look at it, understand, you'll be able to answer. Binding of TNF activates several signaling pathways, including transcription factor, proteases, and protein kinases. Activation of target cells, which are important. What is a target cells? It hits and it annihilates the enemy. Inflammatory and immune response, cytokine apoptic pathway. As you know, the apoptic pathway is the way that it just kills off the nucleus, the nucleosomes, and nucleus cytoplasm. Apoptic, that's one of the ways that the cancer spreads as well. Cancer, mitotic, and all that takes place. So you've got to understand the cytokine and apoptic pathway. And activation of macrophages, T cells, B cells, pro-inflammatory cytokine, and production interleukin 1, interleukin 6, chemokine, IL-8. These are important. They're simple. If you read, you'll understand that. Then I'm going to the antivirals, lopinavir and ritonavir. 199 patients at Jin Yan Tan Hospital, Wuhan, received lopinavir and ritonavir or standard care. Patients on lopinavir and ritonavir showed shorter time to clinical improvement to be included in UK recovery trial. These are again part of the eminent scientists and clinicians working on that. Remdesivir, I call it Gilead Sciences. They seems to be in the pole position. They seems to be doing a lot of trials and being at a steadfastly in a great speed. The ACTT trial, 1,063 hospitalized patients with advanced COVID-19 and pulmonary involvement versus placebo. Interim analysis showed remdesivir 31% faster recovery time. That is significant. 31%, 1 in 3 getting fast recovery time. That's important to the intensive care specialists, especially people like Professor Tahir, Professor Anil Kumar would be delighted to have that kind of rapid, fast recovery. You must know how your peers feel for this kind of medications and treatment. Median time to recovery and remdesivir is 11 days versus placebo 15 days. That's a significant. 4 days is a big change of a patient who is on an acute COVID-19 ward, whether he would go into ventilator, tracheostomy, all the complications or not. Then come mortality rate, remdesivir 8% versus placebo 11% is a big change. So this is something which of keen interest the scientists and the great eminent world scientists are looking at it. Then favipiravir, it's a Glenmark pharmaceutical, which is also another leading pharmaceutical industry which is in the forefront, approved in Japan for new influenza. Phase 3 trial proposal in India to start July, August. 150 patients with mild to moderate COVID-19. Ivermectin. So FDA approved the antiparasitic drug with known in vitro activity against viruses, including HIV, dengue, influenza, and Zika virus. You must know about Zika virus, where it exists in South America, what it's caused, what are the prevalence, and how it's treated is important. It's similar to, you must also read about Ebola virus, but that's not the topic here. Cell culture study at Monash University is being started in Australia. Single dose eradication all viral RNA SARS-CoV-2 within 48 hours. Unknown mechanism against the virus at this stage. Question is that maybe it's dampening down of host cells' ability to clear virus. Hydroxychloroquine and azithromycin. This is something that's been ongoing for the last 6-7 weeks. Initial reports of favorable results. Retrospective cohort study of 1,438 patients in New York. Non-significant versus neither treatment for in-hospital mortality. HCQ 1.08, azithromycin 0.56, HCQ hydroxychloroquine, azithromycin 1.35. ACE inhibitors is again something what San Diego was looking at it. The usage of Ramipril, ACE2